Glycolysis is the process by which glucose is split into 2-pyruvate. 2 ATP are used, being broken down into ADP and phosphate. This is the energy investment phase. The energy return phase is when 2 NADH are produced from 2 NAD+, attaching a phosphate group, which is then used to produce 2 ATP for each 3-carbon sugar, with 4 ATP total being produced in a net gain of 2 ATP. 2-pyruvate are then formed. After glycolysis, the pyruvate can be used either in cellular respiration or in fermentation. We follow this pyruvate onto the aerobic process of cellular respiration. In a transport protein, a carbon breaks off and joins with 2 oxygen to form carbon dioxide. This allows coenzyme A to attach to the 2 carbons and create a compound called acetyl coenzyme A, or acetyl-CoA. Another NADH is also formed. Moving on to the citric acid cycle, the CoA breaks off. The remaining carbon are then transferred to carbon dioxide. During this process, though not all at once as illustrated, 3 more NADH are formed from 3 more NAD+, leaving an additional 3 H+ as well. This yields another ATP from GTP. FAD accepts 2 hydrogen and becomes FADH2, which is used to carry electrons to the next step. In the electron transport chain, proteins carry electrons from NADH. Electrons are eventually passed to the electronegative oxygen, which also picks up 2 hydrogen and forms water. FADH2 is another source of electrons in this process. Hydrogen, pumped against its concentration gradient, comes rushing back into the mitochondrial matrix, spinning a rudder which catalyzes the production of ATP from ADP and phosphate. This process produces about 34 ATP in total. After glycolysis, pyruvate can also be used in fermentation, an anaerobic process. In lactic acid fermentation, pyruvate is used in conjunction with NADH to regenerate NAD+, creating lactate.